Hello everyone, in this video lecture we are going to understand microbial nutrition with respect to their different nutrient types and their transportation inside the cell. Let's start with introduction. Now see, microbial growth conditions. We know that growth is what? So growth is nothing but it is an orderly increase in the cellular constituents, and it is totally depends on the different factors such as first one, that is macronutrients and the micronutrients, and the environmental factors, which includes temperature, pH, oxygen, nutrients, and other factors. On the basis of that, the microbial growth occurs in the environment and in the lab on media. So for that purpose, mostly nutrients are required. So microorganisms require about 10 elements in large quantities because they are used to construct carbohydrate, lipids, proteins, nucleic acids. Several other elements are needed in very small amount and are parts of enzymes and cofactors. Now see, for the microbial growth, the most important factor is the nutrient. And the nutrients are required for the growth of microorganisms because they have to prepare different macromolecules. We say that carbohydrates, lipids, proteins, and nucleic acid. As far as these macromolecules are concerned, they have to take nutrients and they have to metabolize it. But apart from these, they required small nutrients, that is micronutrients also, for the production of different enzymes and cofactors. Now let's start with the microbial nutrition. Now see, what is nutrient? So nutrients are these are the substances present in the environment and used by the microorganisms or organisms for the catabolism and the anabolism. So nutrient is the substance which is present inside the environment and used by the organism or organisms for the catabolism and the anabolism. So catabolism means what? The conversion of complex compound into simpler form is known as catabolism. And anabolism means what? By using that simple form of product, they have to do biosynthesis of nucleic acid or carbohydrates is known as anabolism. And catabolism and anabolism is combinedly known as metabolism. Okay. Our next point is the macronutrients. Now see, these macronutrients are required in large amount to the bacterial cell. For example, carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus. Now phosphorus means what? The components of carbohydrates, lipids, proteins, and nucleic acid. Potassium, calcium, magnesium, iron. These iron like cations and parts of enzymes and cofactors. Now these macronutrients are required to the cell in a very large amount, while the micronutrients are required to the cell very small amount of other mineral elements such as iron, copper, molybdenum, zinc, and these are referred to the trace elements. Most are essential for activity of certain enzymes, usually as a cofactors. So for the growth of microorganisms or any bacteria, the macronutrients and micronutrients are required, while micronutrients required in very large amount, while the micronutrients are required very small amount. Next one, growth factors. Now see, for the growth of bacterial cell or the microorganisms, these are the three important components, that is amino acid, purines and pyrimids, and the vitamins. These three important factors are required for the growth of any organism. That amino acids are required for the protein synthesis, and purines and pyrimids are required for the nucleic acid synthesis. Now vitamins are very small organic molecules that are usually make up all part of enzyme cofactors, and only very small amounts of are required for the growth. Now see, on the basis of nutrient types, the microorganisms are divided in different types. We know that autotrophs means what? The organism those are prepared, like those are able to prepare their own food matter is known as autotrophs. Phototrophs means the organism usually depends on the light, known as phototrophs. Now see, first type is the photolithotropic autotrophy. Now they use light as an energy source, and inorganic hydrogen or electron they use as a hydrogen, that is electron, and they use carbon source, that is CO2, as a carbon source, and the examples are the algae, purple, green sulfur bacteria, and the cyanobacteria. Next is a photoorganotropic heterotropy, means photoorganoheterotropy and the photoheterotrophs. They use light as energy source, and they use organic hydrogen and electron donor, and they use organic carbon as a carbon source. Then purple non-sulfur bacteria and green non-sulfur bacteria are the examples. Next one, chemolithotrophic autotropy, that is chemolithoautotrophy and chemoautotrophs. They uses energy source, that is chemical energy, as energy source, that is inorganic carbon, and inorganic hydrogen, that is electron donor, CO2 as a carbon source. Example, sulfur oxidizing bacteria, hydrogen bacteria, nitrifying bacteria, and iron oxidizing bacteria. Next one, chemoorganotropic heterotropy, chemoorganoheterotropy and chemoheterotrophs. Chemical energy as a source, that is organic source, organic hydrogen, that is electron donor, organic carbon source, that is protozoa, fungi, most photosynthetic bacteria, etc. So these are the some classification based on the nutrients. Now see uptake of nutrients. Now this very important part, the transportation of nutrients which occurs inside the organisms. Now the nutrient molecules frequently cannot cross selectively permeable plasma membrane through passive diffusion and must be transported by one of three major mechanisms involving the use of carrier proteins. That is passive transport, facilitated diffusion, active transport, and group translocation. Mostly the transportation of nutrients inside the cell occurs by these four ways. Let's see, that is passive diffusion. Now a few substances such as glycocerol, H2O, or oxygen can cross the plasma membrane by passive diffusion. Now passive diffusion means what? It is the process in which molecules move from higher region to the lower concentration region. What it is? Passive diffusion, it is a process where the molecules will pass from the higher region to the lower region. Here no carrier proteins are required, means no energy is required in this process. Now we can see this figure where this is a lipid bilayer, and through this lipid bilayer the molecules are coming in. See here the concentration is very high, and this is outside to the cell. So from the outside the molecules are coming inside, that is the higher, from the higher concentration to the lower concentration, is known as passive diffusion. Next one, facilitated diffusion. Now see, name itself indicates that there is a facility to provide or to transfer the molecules from outside to inside or inside to outside. The rate of diffusion across selectively permeable membrane is greatly increased by the use of carrier proteins called permeases, which are embedded in the plasma membrane. Now see, in this diffusion process, whatever the diffusion is going to happen, or whatever diffusion is occurring, that diffusion is carried out by the protein known as permeases. Permeases protein means what? This protein will give the permission for the transportation. Since the diffusion process is aided by carrier, it is called a facilitated diffusion, and this permission is facilitated by the protein known as facilitated diffusion. The rate of facilitated diffusion increases with the concentration gradient much more rapidly and at lower concentrations of the diffusing molecule than that of passive diffusion. Now see, the passive diffusion is the process where the permeases are required to transport the molecule from outside to inside or inside to outside, where this process is carried out by the process known as facilitated diffusion. Now see, we can see this graph, that is the external concentration of soluble to the rate of solute entry. Now this is the simple diffusion, which is at very low, while carrier mediated transport is very high. Because why it is going in very high speed? Because there are permease protein, or there is a facility to transport the protein molecules or transport the any molecule. That's what this process is very high, and this graph is very high. Now see, facilitated diffusion, there is a higher concentration and lower concentration. This one, facilitated diffusion requires carrier protein, that is permeases. Now each carrier is selective and will transport only close related solutes. seem not to be important in prokaryotes, much more prominent in eukaryotic cell. Now see, it is not mostly found in prokaryotes. While see, in the prokaryotes, like prokaryotes mostly unicellular organisms, so it does not require that facilitate diffusion in every time, because it's a very small cell and like everything will pass very easily. But it is important in the eukaryotic cells, as we know that eukaryotes are the multicellular organisms, so it is required there. Now see this see this figure. Now in this figure we can easily observe that, see these molecules are coming inside through one channel, through this channel. So this channel is known as permeases. So this channel is known as permeases. So they are giving the permission to the transportation. Now see this second figure. These molecules are coming in the contact with that facilitated protein, and this facilitated protein gives facility for the transportation, so known as carrier protein. So this carrier protein carries the one molecule from outside to the inside. Next one, active transport. Now it is the process of transportation of solid molecules to higher concentration or against concentration gradient with use of energy input. Here lower concentration, higher concentration, permeases and energy is required. Now see, in this process the molecules are transferred from the higher concentration to the lower concentration, while here energy is required and the permeases are also required. We can see this figure, this is the higher concentration molecule and this is the lower concentration, while these molecules are coming from higher concentration to the lower concentration with the help of ATP. Now here in this process ATP means energy is used for the transportation of molecule. Now this is the proton gradients, nothing but the active transport. Now here we can see first one symport. Symport means what? Linked transport of two substances in the same direction. Symport means what? Two substances are transported in the same direction. For example, water is transferred from outside to the inside, and the glucose molecule is also transferred from the inside to outside. This water and this is a glucose. Now see, glucose and water is transported inside the cell at the same time, known as symport. Antiport means what? The two molecules are transported in the opposite direction. For example, see, antiport means the water is transported to the outside and glucose is transported to the inside. It is known as antiport. Uniport means what? The water is transported in the same side and glucose is also transported in the same direction. Okay, and only one substance will enter inside the cell, that is known as uniport. Group translocation. Now see, this is a process in which molecule is transferred into the cell is being chemically altered. For example, we can take the example of glucose. Now we know that when the glucose is outside to the cell, it is a glucose. When it comes inside to the cell, it becomes glucose 6-phosphate. The chemical structure of original glucose is changed into the glucose 6-phosphate. This is known as chemically change in the structure. So this is known as group translocation, where by the addition of single group inside the or into the original chemical structure is changed. The base known group example for the phosphoenol pyruvate, that is sugar phosphotransportase system, that is PTS. Now here we transfer the variety of sugars into prokaryotic cell by simultaneously phosphorylating them using phosphoenol pyruvate. It is PAP as a phosphate donor. So here we can take an example, that is glucose converts into the glucose 6-phosphate. Now these are the simple comparison of different transport systems. This we will see. Now first one, passive diffusion, facilitated diffusion, active transport and group translocation. Now see, carrier proteins in passive diffusion is not required, facilitated diffusion required, it is required, and here is also required. Transport speed in passive diffusion is very slow because see, here no carrier proteins are involved. That's what passive diffusion is very slow. Facilitated diffusion is very rapid because carrier proteins are involved. Active transport is rapid because carrier proteins are required. Group translocation is also very rapid because carrier proteins are involved. Now it is against concentration gradient. So passive diffusion is not against concentration gradient. It is with. It's along with. That's what it's done. It is also with, but active transport is against concentration gradient. Group translocation is also against concentration gradient. Now passive diffusion transport molecules. Now there is no specificity. They can transport to any molecule. Those are very in the diffusion principle. Passive diffusion, they are specific and they are specific because there is a need of permeases and the facilitated proteins. Metabolic energy, there is no need of metabolic energy because carrier proteins are not required and process is very slow. Here is a need, here is need, and here is need. Means what? The ATP is required. Solid molecules not changed. See, passive diffusion, passive is very simple process. So there is no need to change the solid molecules. Here changed, here changed, and here changed. So this is the simple comparison of transport system in between passive diffusion, facilitated diffusion, active transport, and group translocation. Next one, iron uptake. Now see, cytochromes and many enzymes are uptake by the bacterial cells and the extreme insolubility of ferric iron and its derivatives. Now see, difficult, that is siderophores. We know that siderophores are great examples for the iron uptake. They're like low molecular weight, they contain low molecular weight and they are able to complex with ferric iron and supply to the cell. They forms complex with the ferric iron and they will come inside to the cell. The microorganism secretes siderophores when little iron is available in the medium. Iron siderophore complex bind to the receptor of the cell surface, that is Fe3, release complex entered by ABC transporter. Now see this figure where the receptor is here, okay, and the ferrous 2 plus S. Now with the help of receptor it will come inside, it becomes this one. So this is known as iron uptake. With the help of this receptor, this iron is easily taken by the bacterial cell inside the cell. Thank you.